In this video, we're going to be looking at the adaptive defenses. That is, the ability of the body to develop immunity against specific threats, against specific diseases, viruses, bacteria, whatever. So the adaptive defenses are provided by the actions of lymphocytes and macrophages in response to encountering specific antigens that the body is either infected with or encounters in some way from the environment. And so the two lymphocytes that are involved with immunity are the T cells, or T lymphocytes, and the B cells, or the B lymphocytes. The T cells, they are, like all white blood cells, are produced in the red bone marrow, but before they are completely developed, they migrate from the red bone marrow to the thymus. And there, in the thymus, they fully differentiate under the influence of thymosins, which are hormone-like chemicals that the thymus produces. And then once they are fully developed, fully mature, then they take up residence in the lymphatic organs, especially in the lymph nodes, in the thoracic duct, and in the spleen. The B cells, or the B lymphocytes, we can think of these guys as being bone dependent. So if the T lymphocytes are thymus-derived, then the B cells are bone-dependent lymphocytes, and they differentiate fully and fully develop in the red bone marrow. And then once they are mature, they will take up residence as well in lymphatic organs, and they're found especially in the lymph nodes and in the spleen, but also remain in the bone marrow or can be found in the intestinal lining, among other places. So they're certainly found throughout the body, but those are places where we see especially large concentrations of them. The T cells are activated or trained to recognize specific antigens by a kind of cell that's referred to as an antigen-presenting cell. And there are several different kinds of cells that can act as antigen-presenting cells. Dendritic cells are cells that are found in epithelial tissues. Macrophages can become or can act as antigen-presenting cells. And B cells can also serve as antigen-presenting cells. So an antigen-presenting cell will encounter a antigen or an antigen-bearing particle, and it will phagocytize the particle and then partially digest the antigen-bearing particle. And then, rather than fully digesting that antigen, the antigen molecules are taken and placed on the cell membrane of the antigen-presenting cells. And they are displayed near a particular set of proteins that are referred to as the major histocompatibility complex. So these MHC proteins are found on the membrane, and the antigen-presenting cell will stick these antigens that it's discovered near these MHC proteins. So the antigen-presenting cell then finds a T cell, and it exposes the T cell or the T lymphocyte to the antigen and the major histocompatibility complex proteins. And this serves as a signal to that T cell then to respond to the antigen as if it is foreign. And so now we have an activated T cell that will respond only to antigen-bearing molecules, or I'm sorry, not molecules, but antigen-bearing particles. So a virus that has that antigen or a bacteria that has that antigen will, this T cell is now activated to respond to that. So here is a schematic of how this kind of activation occurs. So here we have a antigen-presenting cell, and it phagocytizes the antigen-bearing particle, and digests away the antigens, and then it can stick these antigens out here on the surface of the cell membrane. And it places them out there near the major histocompatibility complex proteins. Excuse me. And so it will make contact then with a T cell so here's a T cell, or here's another T cell. It'll make contact there, and when the T cell becomes activated, then it will proliferate. So it will begin to divide, producing T cells that will then go out and provide the effect. And so that's where we're headed to next, is to look at the effects then that these activated T cells will have in the tissues. So T cells will interact directly with the antigen or the antigen-bearing particle. So proteins found in the blood could be antigens. So if we have a bacteria, let's say, and it's producing a toxin in the blood, then that toxin may act as an antigen, or the bacteria will be the antigen-bearing particle. So T cells will interact directly with these antigen-bearing particles and will secrete chemicals referred to as cytokines that will help to help the T cell to really, and other T cells to really respond to this antigen. So what are some of those cytokines? So interleukin-1 and interleukin-2 are two of the cytokines that they can produce. So if the T cell encounters an antigen-bearing particle, then it could release interleukin-1, which will help to activate other T cells and also stimulate them to produce cytokines. Interleukin-2 will cause the T cells to reproduce, so that we get more T cells that are activated to this antigen, and also to cause those reproducing T cells to produce cytokines as well. Colony stimulating factors are cytokines produced by activated T cells that help to encourage the bone marrow to produce more leukocytes, to produce greater numbers of white blood cells. And then tumor necrosis factor is a chemical that an activated T cell can produce that helps to stop tumor growth, as well as stimulate the growth and differentiation of lymphocytes. Excuse me. This tumor necrosis factor also can act as a pyrogen in the body to cause fever to be produced. So we can produce the T cells can produce cytokines in response to encountering an antigen that it's activated to, and it can also directly interact with these antigens or the antigen-bearing particle and secrete toxins like perforins that will cause poke holes essentially in the cell membrane of the antigen-bearing particle, then rendering it susceptible to osmotic stress, so that helps to kill the cell. It can secrete growth inhibiting factors that will slow the growth of the cells that have these growth inhibiting factors secreted onto them. And they can also secrete interferons that will inhibit virus and tumor cell growth. So not only will a virus-infected cell produce interferons, but if a activated T cell encounters its antigen-bearing particles, then it will also produce interferons. So we talk about the action of T cells as being cellular immunity, because these T cells interact directly with the targeted antigen-bearing particles to remove them from the body by either lysing them, slowing their growth, or producing interferons that are going to inhibit or or the growth of a virus within within nearby cells. Okay, so T cells provide cellular immunity to remove the targeted antigen-bearing particles. So what are the types of T cells that we make? So there are cytotoxic T cells, and these are T cells that can bind to and then directly attack a cell that has been identified as cancerous or as virus infected. And they do this by secreting perforins that will help to lyse the cancer cell or the virus infected cell. So here we see a cytotoxic T cell. Here is a cancer cell that it's interacted with, and you can see the holes that the perforins have made in that cell. Okay, so that's a cytotoxic T cell that helps to destroy a cancerous or virus-infected cell. There are also helper T cells. Helper T cells are helpers because they are going to be, we're going to see in a minute, that they're involved in stimulating B cells to produce antibodies. And the HIV virus is particularly virulent because it specifically targets these kind of helper T cells. So helper T cells are going to be involved in activating B cells to get them to produce antibodies. And then third are memory T cells. So as we get, as T cells interact with their antigen-bearing particles, they will divide, and some of the offspring cells that are produced are termed memory T cells because they will remain in the body, and if they later, after this event is over, if they encounter that same antigen again, then they will divide immediately and produce a cytotoxic T cells and helper T cells that will then be involved in fighting that infection that's reoccurred. Oh, I forgot to point out this image here. I love this one too. The helper T cell is in pink, and it is this is a helper T cell that has been infected with the HIV virus, and you can see the green HIV virus particles that are budding off of the helper T cell as it gets sort of unbeknownst to the helper T cell, it's now been recruited. Rather than fighting the HIV virus, it is actually spreading the HIV virus. And again, that's a big piece of why the HIV is such a damaging infection, is because it targets the immune system directly. Okay, so those are the T cells that do the cellular response. Provides cellular immunity by attacking cells of self cells that have become cancerous or have been virus infected. Okay, so now let's look at the B cells. So when a B cell is activated, its job then is to produce antibodies, and these are proteins that will bind to the antigens that that the B cell has been tuned to recognize, and it will signal then to other cells that, hey, we've found something that's non-self, we've identified it before, it's time to get it out of here. So B cells are activated by helper T cells that have been sensitized to the same antigen as the one that the B cell has been activated by or sensitized to. So as we saw with T cells, antigen-presenting cells will present to B cells, and these B cells then are going to be sensitized to that antigen. But when they then encounter a helper T cell that is also sensitized to that same antigen, then the helper T cell will produce cytokines that it squirts all over the B cell that cause the B cell to divide. And so it begins producing clones of itself. And these clonal daughter cells can differentiate into one of two kinds of cells. if it differentiates into a plasma cell, then these plasma cells are the ones that produce the antibodies that will bind to antigens, and as it makes these antibodies, it will secrete them into the blood or into other body fluids as well. So it's not just blood that carries the antibodies. As the B cell divides, it may also produce or throw off some memory B cells. And like the memory T cells, the memory B cells are initially inactive, but when they encounter the antigen later on, they will divide immediately and produce plasma cells and more memory B cells. So the memory B cells are there, and the value of having these memory B cells is that you don't need a helper T cell. You don't need that double check off to start making antibodies against something. And so this really, really speeds up the process of getting more antibodies out into body fluids if you don't have to go through the process where first a B cell gets activated, and then in the same area helper T cells get activated, and then they find each other, and then the helper T cell stimulates the B cell to divide. You don't have to go through all those steps. Now, once there are memory B cells available in the body, then they can immediately go divide and start making plasma cells that will immediately begin producing antibodies with no wait time at all. So an adult has somewhere between 10 million, and this is just amazing to me, and 1 billion different types of B cells. So I'm not talking about different B cells, I'm talking about different types of B cells that have been sensitized to a particular antigen. So each one of these 10 million to 1 billion types is sensitized only to one antigen. And so as the plasma cells, the offspring plasma cells of the B cells, really crank up the production of antibodies, they can produce as many as, one plasma cell can produce as many as 2,000 antibody molecules every second. So again, individual plasma cells make only one antibody type, but a pathogen, so if we are thinking now of a bacteria, let's say, it has multiple antigens on the surface of its membrane. And so when we start activating B cells, some will be activated to this kind of this kind of identifier protein. Some will be activated to this one, some will be activated to this one. So the immune response that we get is to be able to bind antibodies to this type of protein, to this type of protein, and to this type of protein. So we describe the immune response as a polyclonal response because we make antibodies from a set of clones of plasma cells that are activated to, let's say, the circular-looking antigen, and a whole nother set of clones that are activated to this square-looking antigen, and yet a whole nother set of clones that are activated to this triangular antigen. So it's a polyclonal response, and we can really tack on to the membrane now of the bacterium a bunch of different antibodies so that we really get a great strong signal out there that, hey, this cell has been identified by the immune system as not belonging to the body. So we describe the type of immune function that's associated with the B cells as humoral defense because the antibodies are found in body fluids. Back in the olden days, like Shakespearean times, the humors were talked about as the fluids of the body, so blood and the contents of the stomach are another humor, and urine and phlegm, mucus fluids. All of these are different humors of the body. And because antibodies are found in body fluids, then we talk about this as the humoral immune response or the humoral defense. And just now, to wrap up our understanding here, we have a B cell that has been activated against a specific antigen, and it was activated. And if it gets cytokines from its helper T cell, that's also activated to the antigen. And if it encounters the antigen out there, so now we've got all these things that have happened. It is activated to an antigen, it's encountered the antigen, and there's a helper T cell nearby that squirts cytokines all over it, then it will proliferate. It will begin to divide, and we'll get more and more of these B cells. So we'll get these monoclonal B cells that are produced, and they will proliferate as well. They will divide and differentiate, and some of them will become memory cells, and they're initially dormant, but if we encounter that antigen later on, then they will divide and produce plasma cells. And some of those cells will become plasma cells. And when these plasma cells are produced, then they will immediately begin producing antibodies that it will release then into the fluids. And so here you can see the antibodies being released. Here are some antibodies being released out into the body fluids after they get made. All right, so there you have it. Activation of T cells stimulates the cellular response, and we can start killing our own cells that have become virus infected or are producing cancers. Or, with the assistance of helper T cells, the B cells will initiate the humoral response, and we can see division of the B cell to produce plasma cells that will then produce antibodies that can go and bind to the antigen and serve as a signal to other cells that, hey, we need to come and remove this antigen-bearing particle. Okay, so we're going to look then at the action of antibodies in the next video, but that's it for this one. So you know how to reach me if you have any questions.